And I will show 3 examples that we received from actual users of this graph database. So people who approached us and said that these are the questions I have for my research. So the first is, can I find all the small molecules in the database that have the same scaffold and that interact with the same binding sites? And this is something you can quite easily do in this graph database. And in fact, you can add additional information such as, are these binding sites catalytic sites, or the small molecules, are they drug-like, are they cofactor-like, reactants? How conserved are these shared binding sites? And much more than that, actually. So if you were to look at the graph, this is what you would see. You have one isoleucine here, which is actually a residue from UniProt. And you can see that this particular residue is represented in 2 PDBe residues. So basically there are 2 PDBe entries that have this isoleucine. And in the graph, you can see that actually both of them have connections to a small molecule, different small molecule, and these 2 happen to be antibiotics. And if you look at those small molecules, you can see that actually they are similar, which is based on several criteria. One of them that you can filter for is that they have the same scaffold. So this is already visible if you just look at the graph database. But if you actually look at the examples, you can see that indeed, these 2 antibiotics, they have the same scaffold. And if you were to visit one of these aggregated pages, aggregated views that I will talk about later, you could go to the ligands section and you could see which residues of the protein are interacting with which small molecule. So here on the x-axis, you can see the residues of the protein. And these blue boxes, they represent the residues which are interacting with a particular small molecule. And so you can see that basically there is only one residue difference in interaction between these 2 small molecules. Another example that we received was, can I find all the macromolecular interaction interfaces where the residues are both highly conserved, but also predicted to be structurally disordered or flexible? And again, yes, this is doable with the database. And in fact, you can add information like what domains are mapped to these residues, or do these residues fall into a specific type of secondary structure element? And if you were to look at the actual graph, the way it looks like is, it will come up with the groups of amino acids. And on one of the edges, you would find the value for the conservation, for the sequence conservation. You would also find on their actual node if they are predicted to be disordered or not. So this information is coming from a resource called MobiDB. And also you could see that actually it interacts with another protein. So it is on interaction interface. This interaction data is coming from PISA. And the third question that I would like to show today is about variants. So is it possible to get all the residue pairs from the complete PDBe archive, where there is a residue and in one structure it's the wild type, and in another structure it's some kind of variant, but the same exact position of the protein? And this is also quite straightforward actually to do in the graph. And you can enhance this data set with information on are these residues involved in interaction interfaces? Are they interacting with small molecules? And in fact, if you are interested, you can dig down to the atomic level details of small molecule interactions. Are these residues conserved? Are they buried, accessible? How accessible they are in terms of angstrom squared? And are these flexible? And so on. Actually, there are much more in there. If you were to look at a specific example, you could see this. For example, we have a glutamine here, and this is the UniProt residue. You can see that all these blue circles, those are from different PDBs where you actually have this UniProt residue. And you can see that in the majority of the cases, this is a glutamine, but there are a few cases where it's not. And one of the cases is it's an aspartic acid. And I just zoomed on this particular residue because then you can see what information we actually have for that particular node. And so, for example, here we have information on backbone flexibility, for example, or is it an early folding residue? Or I think this is about the interaction interface size, if it has any. Then we have domain annotations from Interpro, Scope, CathB, Cath, and also we are directly mapping this residue to Ensemble as well. Additionally, it turns out that this particular aspartic acid is involved in interaction with a small molecule as well. These are a few examples of what you can do with the graph. And in fact, if you are interested in more, you can just go to this location. It's a Google Drive folder where you can find more materials. You can find the actual queries that you could write and would return these results for you. But there are much more in there as well. We also have a tutorial that you could visit and learn how to retrieve the database, how to set it up. Actually, it's quite straightforward to set this up, and how to write queries. You can also look at the database schema, which actually tells you what exactly we have in the graph and what nodes and what edges are there. And like I said, you can download the complete database, which is refreshed every week. So every week it's up to date.